I want you to imagine that you're standing at the base of this towering state-of-the-art skyscraper.
Okay, like a really modern high-rise.
Right, exactly. And the steel beams are just perfectly forged. The concrete scaffolding is rock solid. And, the glass facade is seamlessly deflecting the wind. It looks permanent. Invincible, even.
Which is, that's the illusion of all great architecture, right? We just assume the structure is totally static once the building is finished.
Yeah, but deep inside the walls of this particular building, something really alarming is happening. The thick steel beams and the reinforced concrete holding the entire thing up, they suddenly start dissolving.
Oh, wow.
And they aren't dissolving because of like an earthquake or some outside demolition crew. They're dissolving from the inside out. They're being broken down by microscopic molecular machinery that the building itself actually manufactured and unleashed.
It's such a striking visual, and it really captures the physiological reality that we're dissecting today. Because, well, that building is you. That structural collapse, it's happening on a cellular level during muscle atrophy. We tend to think of our muscles as these fixed anatomical features, but they are constantly being built up and torn down.
Okay, let's unpack this, because today's deep dive is focusing specifically on the extracellular matrix, the ECM, and these biological enzymes known as matrix metalloproteinases, or MEPs.
Right, the MMPs.
Yeah. And if the ECM is the steel and concrete scaffolding of our tissues, the MMPs they're these specialized enzymes that actively degrade this matrix when tissues shrink or adapt or just completely fail.
Exactly. And our focus today is really isolating the exact role these MMPs play in that degradation process. We're looking at the molecular signaling that tips the scales from healthy tissue maintenance over to rapid structural destruction.
Especially during periods of disuse or severe mechanical stress, right?
Yeah, exactly.
So before we can even understand how muscles break down, we first need to look at the scaffolding itself. The extracellular matrix isn't just some inert biological glue holding our cells together.
Far from it.
It's this highly dynamic three-dimensional web made of structural proteins like collagen and elastin. And it literally dictates tensile strength, elasticity, and even biochemical signaling for the cells.
And because it is so dynamic, the body needs a mechanism to remodel it constantly. You can't reshape a tissue without breaking down the old framework first.
Right, you got to clear the rubble.
Exactly. And that is the primary domain of the matrix metalloproteinases. Biochemically, MMPs are zinc-dependent endopeptidases.
Which means they need zinc to work.
Yes. They require a catalytic zinc ion right at their active site to function. And they are uniquely capable of cleaving the peptide bonds within those incredibly tough ECM proteins.
Especially fibrillar collagen, right?
Yeah, fibrillar collagen is their main target.
Which actually brings up one of the most bizarre and honestly fascinating historical footnotes I've ever seen in our sources. Scientists first isolated these enzymes not in human muscle, but in the tails of tadpoles.
Wait, tadpoles. Yeah, it sounds absurd until you consider the biology of metamorphosis. A tadpole has to completely resorb its tail to become a frog. That requires just massive, rapid tissue degradation. So the tadpole utilizes really high concentrations of MMPs to dismantle its own tail scaffolding.
So the underlying amino acids can be repurposed for the frog body.
Exactly.
Okay, so it makes sense in a tadpole. And, it makes sense during embryogenesis or angiogenesis, or even if you get a deep laceration and the body needs to clear away damaged collagen to build fresh tissue.
Sure, normal wound healing.
But here's where I kind of get hung up. If these zinc-dependent endopeptidases are that efficient at chewing through our vital structural proteins, why do we tolerate them constantly circulating in healthy tissue?
It's a great question.
It sounds like keeping a live biological grenade in your pocket.
Yeah.
Like they're the ultimate demolition crew equipped with zinc-powered scissors.
What's fascinating here is the biochemical failsafe the body actually employs to prevent that indiscriminate destruction. MMPs are almost never secreted in an active state. The cells synthesize and release them as latent zymogens. We specifically refer to them as pro-MMPs.
So they're circulating, but they're functionally locked.
They are completely blocked. The pro-MMP structure includes this specific propeptide domain that folds over and binds directly to that catalytic zinc ion we mentioned earlier.
So it just covers up the scissors.
Exactly. As long as that propeptide sequence is attached, the enzyme absolutely cannot interact with the extracellular matrix. For the MMP to become active and actually start cleaving collagen, that propeptide domain has to be physically severed by other proteolytic enzymes out in the extracellular space.
Okay, that answers the safety question. The grenade is pinned, but that immediately leads to the next variable, what pulls the pin.
Right.
If someone breaks their femur and their leg goes into a cast, the skeletal muscle rapidly atrophies.
Yeah.
Does the sudden lack of movement just trigger a massive release of these proteolytic enzymes to unlock the MMPs?
You would assume it's a direct universal cascade like that, but the physiology of disuse is actually far more nuanced. The mechanism of atrophy heavily depends on the specific nature of the mechanical unloading.
How so?
Well, there is compelling genomic data comparing two distinct models of muscle disuse in mice. They looked at rigid hind limb casting versus tenotomy.
Tenotomy being the surgical severing of the Achilles tendon.
Right, so you have one model where the calf muscle is locked in a cast but still intact, and another where the muscle is free to contract but completely detached from the bone.
So it can't bear any mechanical load at all.
Exactly. Now, the clinical endpoints at day 14 were identical. The researchers measured the exact same reduction in muscle wet weight, the same decrease in fiber cross-sectional area, and the same loss of force production in both models.
So visually, macroscopically, the atrophy was completely indistinguishable.
Completely. But the genome-wide expression profiling told a totally different story about how the cells actually achieve that shrinkage.
Wait, so the body achieves the exact same result, a shrunken muscle, using two entirely distinct sets of tools depending on how the injury happened.
Yes. The divergence at the molecular level was staggering. In the casting model, the degradation was heavily driven by the ubiquitin proteasome system.
Okay, remind me how that works.
It's this highly targeted intracellular pathway where E3 ubiquitin ligases basically tag specific myofibrillar proteins for destruction by the cell's internal proteasomes.
Got it. So it's an inside job.
Right. But the tenotomy model barely activated that pathway at all. Instead, the severed tendon triggered lysosomal degradation and a massive upregulation of our zinc-dependent enzymes, the MMPs.
Wow.
The cells were essentially using autophagy, literally eating their own internal and external structures.
Wait, I need to push back on this. If the biological endpoint is a smaller, weaker muscle in both scenarios, why would the body utilize two entirely distinct sets of molecular machinery just because the method of unloading changed?
That's a fair question.
Like, what difference does a severed tendon make compared to a rigid cast?
It all comes down to mechanotransduction, which is how cells convert physical forces into biochemical signals. In a cast, the tendon is intact, meaning there's still resting passive tension on the sarcolemma.
The muscle cell membrane.
Right. The mechanosensors detect that passive tension and activate the ubiquitin proteasome pathway. But in a tenotomy, that passive resting tension drops to absolute zero.
Because the tendon is cut.
Exactly. The mechanosensors register a catastrophic loss of load, which triggers a much more aggressive systemic remodeling response, utilizing the MMPs to tear down the extracellular matrix itself.
So there's literally no single atrophy switch in human biology.
None. And this implies something huge for clinical therapeutics. If we treat a patient immobilized in an ICU bed the exact same way we treat a patient recovering from a ruptured patellar tendon, we might fail completely.
Because we'd be targeting the wrong pathway.
Exactly. Combating muscle loss requires targeting the specific molecular pathway, whether it's E3 ligases or MMPs, that the specific mechanical environment actually triggered.
Okay, so let's focus on the scenarios where the MMPs do take the lead in driving the atrophy. How exactly does breaking down the scaffolding outside the cell force the muscle fibers inside to shrink?
Well, we have to look at non-weight-bearing disuse to understand that.
Right, like the 14-day rat tail suspension model from the sources, which mimics microgravity for astronauts or prolonged bed rest.
Right. By suspending the hind limbs, you completely remove the mechanical loading from postural muscles like the soleus. And as we'd expect, based on that loss of tension, the genome profiling in those atrophying soleus muscles showed a highly significant spike in the expression of MMP2.
So the local fibroblasts and muscle cells were just flooding the extracellular space with these enzymes.
Yes, but the accelerated destruction wasn't just caused by an increase in MMP2, it was compounded by a simultaneous drastic drop in TIMP2.
TIMP2 being the tissue inhibitors of metalloproteinases.
Exactly. The TIMPs are the endogenous regulatory brakes on the MMPs. So if the propeptide domain prevents the enzyme from activating in the first place, TIMPs are the molecules that bind to and neutralize MMPs that are already active in cutting.
Okay, so muscle atrophy isn't just a case of hiring too many workers for the demolition crew, the MMPs. It's also about someone cutting the brake lines on their bulldozers.
That is the perfect analogy, yes. You're pressing the accelerator and cutting the brake lines at the exact same time. Tissue health is entirely dependent on the stoichiometric ratio of MMPs to TIMPs.
The MMP to TIMP balance.
Right. When that ratio tilts, the balance of extracellular matrix turnover spirals out of control, leading to pathological degradation. In the atrophying soleus, unchecked MMP2 aggressively cleaved collagen auto and collagen IV.
And collagen that feeds the primary structural component of the basal lamina, right? That specialized layer of the ECM that directly wraps around every single muscle fiber.
Yes, exactly. So by breaking down the basal lamina, the MMPs are literally dissolving the immediate interface between the cell and its environment. And this answers your question about how external breakdown causes internal shrinkage.
Right. How does the outside affect the inside?
The sarcolemma of the muscle cell connects to the basal lamina through these transmembrane proteins called integrins.
So they anchor the cell to the scaffolding.
They do, but integrins do way more than anchor. They actually transmit survival signals into the cell. When MMPs chew through the collagen IV, those integrins lose their physical connection to the matrix.
Oh, wow.
So the cell membrane becomes physically compromised and highly permeable, but more importantly, the integrin-mediated signaling just collapses.
The cell realizes the scaffolding is gone.
Exactly.
Yeah.
The cell senses that it has lost its scaffolding, which triggers downstream catabolic pathways inside the cell, driving the reduction of myofibrillar proteins and ultimately shrinking the muscle fiber.
Man, it's basically molecular sabotage.
Yeah.
The broken scaffolding essentially commands the cell to start shrinking. And the data noted that this intracellular MMP2 activity was highly localized to type 2 muscle fibers, right?
Yes.
The fast twitch fibers that are typically the first to degrade during severe disuse or like microgravity.
Which really highlights how targeted this process is. The fast twitch fibers, which rely heavily on explosive mechanical tension, they're the most sensitive to the loss of that tension and the subsequent loss of integrin signaling.
Okay, here's where it gets really interesting. We've mapped out how a loss of mechanical tension unleashes MMPs in skeletal muscle in our limbs. But skeletal muscle gets to rest. Cardiac muscle does not.
Never.
So what happens when this exact same ECM degradation targets the most vital muscle in your body, the heart? What happens when this identical MMP-TIMP imbalance strikes the myocardium?
Well, the microscopic signature is incredibly consistent. When a patient develops congestive heart failure, we see a massive aberrant activation of MMPs in the cardiac tissue. They essentially break the ties that bind by damaging the myocardial collagen.
But wait, a failing heart isn't experiencing disuse, right? If anything, it's working harder than ever to pump against high blood pressure or damaged valves. So what triggers the MMP release there?
So in the myocardium, the trigger isn't a loss of tension, it's pathological stretch.
A stretch.
Yeah, heart failure often involves volume overload. The chambers dilate and the cardiac fibroblasts are stretched far beyond their physiological limits. That chronic, agonizing mechanical stretch causes the fibroblast to overexpress MMPs.
And those MMPs just start attacking the myocardial collagen.
They systematically cleave the fibrillar collagen network that tethers the cardiac myocytes together. So the heart literally physically loses its structural geometry. The walls thin out, the chambers dilate even further, and the contractile force just plummets because the cells no longer have a rigid matrix to pull against.
Wow. So in the leg, zero tension causes MMPs to destroy the matrix. But in the heart, extreme stretch causes MMPs to destroy the matrix.
Exactly.
And to figure out the cardiac side, the sources talked about researchers utilizing a heterotopic heart transplantation model, essentially transplanting a donor rat heart into the abdomen of a recipient rat.
It is an incredibly elegant way to study mechanical unloading in cardiac tissue.
So how does it work?
Well, the recipient rat's native heart continues pumping blood systemically, but the transplanted heart is vascularized and beating, but it isn't pumping against the systemic vascular resistance. It is completely mechanically unloaded.
It's basically the cardiac equivalent of the tail suspension model. The heart is basically in zero gravity.
Right. And the findings in the unloaded myocardium mirrored the skeletal muscle models in terms of activating atrophic pathways. The unloading triggered the mTOR pathway and the upregulation of those muscle-specific ubiquitin ligases we talked about, specifically atrogen-1 and MER-F1.
But translating the concept of cardiac unloading to human patients is where I found a massive contradiction in the literature. Because in clinical practice, we unload failing human hearts using left ventricular assist devices, or LVADs.
Right, mechanical pumps.
Yeah, these are implanted mechanical pumps that take over the workload of the left ventricle. So the LVAD mechanically bypasses the failing ventricle, drastically reducing the volume and pressure load on the stretched cardiac tissue.
It essentially unloads the failing heart.
Right. But hold on, let's look at the mechanics here.
Yeah.
In our skeletal muscle, when we unloaded the soleus muscle by suspending the rat, MMP2 spiked, TIM2 dropped, and the basal lamina was destroyed.
Yes.
But when researchers analyzed human myocardial tissue before and after LVAD support, unloading the heart did the exact opposite.
It is a phenomenal paradox, isn't it? Mechanical unloading of a failing human heart with an LVAD actually induces a significant downregulation of MMPs.
It goes down.
It goes down. The proteolytic activity drops, the MMP-TIMP ratio actually normalizes, and the degradation of myocardial collagen halts.
I am genuinely struggling to reconcile this. How can the exact same physical stimulus, taking the mechanical load off a muscle, cause MMPs to attack in the leg, but cause MMPs to retreat in the chest?
I know, it seems completely contradictory. But if we connect this to the bigger picture, the contradiction resolves when you look at the baseline physiological state of the mechanosensors.
Okay, lay it out for me.
A healthy skeletal muscle requires a baseline level of mechanical tension just to maintain homeostasis. When you unload it, you drop the tension below the physiological baseline. The cells interpret that as disuse, triggering MMPs.
Ah. But the failing heart isn't starting at a healthy baseline.
Exactly. The failing heart is existing in a state of severe pathological tension. The fibroblasts are chronically overstretched, which is the exact stimulus driving the MMP overexpression in the first place.
Oh, I see.
When the LVAD takes over the pumping, it isn't inducing disuse below a healthy baseline. It is simply removing the pathological stretch. It returns the tension to a normal physiological level.
So the LVAD basically tells the overstretched mechanosensors, hey, the pressure's off, you can stop producing the demolition enzymes.
And the cells respond immediately. The pathological remodeling halts. It proves that extracellular matrix degradation is not an irreversible one-way street.
Wow.
By manipulating the mechanical environment, we can actively push the MMP-TIMP ratio back into equilibrium.
So what does this all mean for you listening? Whether we are talking about a tadpole absorbing its tail, an immobilized calf muscle shrinking in a cast, or a failing heart expanding under pressure, the underlying architecture of life is defined by this constant push and pull.
It's an incredibly dynamic system.
The extracellular matrix is a highly volatile environment, and it is governed by a strict mathematical ratio.
Right. The zinc-dependent matrix metalloproteinases do the cutting, and the tissue inhibitors of metalloproteinases act as the brakes. When you alter the mechanical forces on your body, you are directly manipulating that cellular math.
Every single time you push through a heavy workout and create micro tears in your muscle, or every time you spend a week sedentary on the couch recovering from an injury, you are actively altering the tension on those cellular integrants.
You are.
You are quietly commanding this invisible, microscopic battle of proteins to either reinforce your scaffolding or tear it down.
Our biology is exquisitely sensitive to the mechanical demands we place on it. The molecular machinery is always, always listening to the physical forces.
Which leaves us with a really provocative frontier to consider for the future.
Oh, definitely.
We know that altering mechanical stress, like implanting an LVA, can normalize the MMP-TIMP ratio and halt cardiac collagen degradation. We also understand the precise molecular structure of the TIMP proteins that jam the MMP's active zinc sites.
They do.
So if we can map all of this, could medical science eventually cross the threshold into direct pharmacological manipulation?
Well, developing a selective synthetic MMP inhibitor that doesn't completely disrupt normal biological functions like wound healing is one of the most complex challenges in molecular pharmacology right now.
But imagine the implications if we succeed. Could we administer a targeted therapeutic that temporarily locks the molecular scissors in a patient suffering from severe spinal cord injury, completely arresting their muscle atrophy while they are bedridden?
That would be revolutionary.
Or could we engineer a pharmacological brake line for astronauts embarking on multi-year space flights in zero gravity, ensuring their basal lamina remains fully intact without relying solely on exercise machines? If we can master the molecular cues that command the demolition crew, we might finally learn how to make the skyscraper scaffolding truly invincible, no matter what environment we put it in.